Hello everybody. First of all, I'd like to thank Dr. Ben Albensi for giving me this opportunity to present my work in the Dementia Journal Club. Today I'll talk about how transferrin trafficking, turnover, and iron homeostasis are regulated by a calcium-activated kinase signaling pathway involving two kinases, CAMKK2 and its downstream kinase CAMK4. This presentation is based on some of my last 2 years' research activity, which is published in 2 journals. The Frontiers in Molecular Bioscience paper was published in November 2018, already received 6 citations. One paper recently accepted in Cell Communication and Signaling, which is now in press. Both are open access journals, so the papers can be downloaded free. Before I go into the details of my work, I like to present a video abstract of my Cell Communication and Signaling paper, which will be made available in the web version of this journal. This will help the audience to have a brief overview of this presentation. In this presentation, I designed this lecture student-oriented. Based on my past teaching experience, I thought that it would be wise to give you some background concepts and information on the topics, which may help to digest the work more easily. I'm sure most of you are aware of these basics, but please be with me. I have written some learning objectives which will guide you through my lecture. First, I'll discuss about different hypotheses of Alzheimer's, specifically the calcium and metal ion hypothesis, the properties of calcium that makes it suitable as a signaling molecule, role of iron, how calcium signaling works, and how iron is transported in the body. Next, I'll discuss paper 1, which mainly describes how CAMKK2 was discovered as a regulator of transferrin trafficking, and this discovery led to the identification of phosphorylated transferrin as a potential biomarker for Alzheimer's disease. This discovery has been protected by an IP filing through University of Manitoba, and based on that, I formed a startup company named Alzo Bioscience Incorporation to bring this technology from bench to clinic, specifically for Alzheimer's disease diagnostics. Next, I'll discuss about paper 2, which is identification of CAMKK2 downstream kinase CAMK4 as a co-regulator of transferrin trafficking, and I'll provide you evidence of CAMK4 loss and iron dyshomeostasis in the brain. Then I'll discuss about how CAMKK2 CAMK4 signaling regulates endoplasmic reticulum mediated calcium release response during cholinergic signaling, and also how altered calcium homeostasis is associated with defective transferrin trafficking. I'll also show you some interesting data that shows CAMKK2, CAMK4 signaling regulates the dynamics of transferrin and transferrin receptor associated protein complexes during trafficking. Alzheimer's disease is characterized by two cardinal pathological hallmarks: the neurofibrillary tangle, which is an abnormal accumulation of phosphorylated tau within the pericarial cytoplasm of certain neurons, and the senile plaque, a central core of beta amyloid protein, a 4 kilodalton peptide surrounded by abnormally configured neuronal processes or neurites. In this Belchowski stent and phosphotau immunohistochemical stent human Alzheimer's patient's temporal cortex section, you can see the presence of both NFT and senile plaques. Over the years, multiple hypotheses have been proposed for Alzheimer's disease, but not all are capable of explaining the basic pathological changes of Alzheimer's. The amyloid hypothesis has been the mainstream concept underlying Alzheimer's research for over 20 years. However, the fact that there are many healthy people with amyloid deposits and Alzheimer patients with very few amyloid deposits, and the failure of the drugs targeting amyloid deposits in clinical studies suggest that alternative hypotheses are necessary to explain Alzheimer's. In particular, the calcium hypothesis, that is the pathogenic role of altered calcium in Alzheimer's, has become more prominent in recent years. In this figure, you can see that different hypotheses are listed with a percentage of clinical trials that has been based on these different hypotheses. The calcium hypothesis is very interesting because it is also connected to the amyloid hypothesis and neurotransmitter hypothesis. On the other hand, the metal ion hypothesis is also related to mitochondrial cascade and related hypotheses. The calcium hypothesis suggests that altered neuronal calcium is associated with progression of Alzheimer's disease. It was first proposed in 1992 by Matson et al. It has been shown that amyloid beta can elevate intracellular calcium levels. Calcium homeostasis is closely related to learning and memory. The metal ion hypothesis suggests that different metal ions are associated with Alzheimer's disease, for example, iron, copper, and zinc. Iron is required to act as a cofactor for enzymatic activity, mitochondrial function, and neuronal function. During normal aging, it has been found that there is gradual accumulation of iron in different parts of the brain. But in Alzheimer's brain, there is a dramatic increase of brain iron content. An increase in the level of iron in AD brain was first demonstrated in 1953. Iron can reach high concentration, approximately 1 millimolar in the plaques. Iron overload accelerates neuronal amyloid beta production and consequently worsens cognitive decline in a transgenic AD mice. Calcium is one of the most ubiquitous signaling molecules. Before discussing the properties of calcium that makes it suitable as a signaling molecule, I like to highlight the relative abundance of calcium, iron, and magnesium in the universe, as well as in the human body. Iron and magnesium is more abundant in the universe compared to calcium, whereas calcium is more abundant in the human body compared to magnesium and iron. Interestingly, magnesium is the 8th most abundant element in the Earth's crust and the 4th most common element in the Earth. Now, both calcium and magnesium are essential minerals and critical components of healthy bones and teeth. Both belongs to the second column on the periodic table of the chemical elements, collectively known as alkaline earth metals. And because of the outer electron structure in calcium and magnesium, they all have similar chemical properties and physical properties. Therefore, it is reasonable to hypothesize that there must be some reason behind the choice of calcium as a signaling molecule in the living organism. In the next slides, I will highlight some of the chemical properties of calcium that makes it more favorable as a signaling molecule compared to iron and magnesium. There are multiple factors that affect interaction of metal ions with coordinating ligands. In proteins, metallic ions are surrounded by molecules or ions of the surrounding amino acids of the protein that are known as ligands or complexing agents, and together they form a coordination complex. In calcium and magnesium, both has 2 electrons in their outermost shell, hence its valency 2. That means it will lose 2 electrons to achieve noble gas configuration. On the other hand, iron combines with oxygen to form ferrous oxide as well as ferric oxide. In the formation of ferrous oxide, iron exhibits a valency of 2, whereas in ferric oxide it has a valency of 3. The next factor is the ionic radius. The ionic radius is important because it shapes the electron cloud of the ion, which is important because the ion has to fit within a pocket of the metal binding site in the protein. Thus, a certain ionic radius is more favorable than others. For example, the ionic radius of calcium is 0.99 angstrom, whereas the magnesium ion 0.65, ferrous 0.74, and ferric 0.64. The next factor is the polarizability, the ease with which electron cloud of the metal is distorted by external electrical forces. It is important to note that the outermost electrons in the metal ions interact with electrons in the ligand molecule or atom in a coordination complex to form a stable structure. This interaction involves atomic forces, for example hydrogen bonding, van der Waals force, or covalent bonding, etc. And all of them involves manipulation of the electron cloud in the metal ion. Therefore, calcium with a high polarizability may be more favorable to form coordination complex in the protein. 2 other factors that favors calcium over magnesium as a signaling molecule. These are the hydration energy and the radius of the hydrated ion. The hydration energy is the ease with which the attached water molecules are stripped of the metal ions. Due to their charge, the metal ions are surrounded by polar water molecules in an aqueous environment of the biological system. For example, magnesium is surrounded by 2 hydration shells, whereas calcium has just 1 layer. We know that the metal ions bind with proteins and cause conformational change, which then act as a signaling cue or signaling molecule. But another important criteria to work as a signaling molecule is that the metal ions have to be transported across biological membranes to convey signals. Now, if calcium or magnesium needs to be channeled through the membrane-bound ion channels, they have to be first stripped of their hydration shells during the transport process. Because most of the membrane-bound ion channels has a specific diameter which is necessary for their ion selectivity. Thus, those ions are favorable that has less hydration energy. Therefore, calcium is more favored because it shades its single hydration shell and its dehydrated ion easily fits the ion channel. Magnesium, on the other hand, has to get rid of 2 layers of water, which is highly energy consuming and thus less favored compared to calcium. Calcium exists in at least 3 basic forms. Number 1, ionized. Number 2, complex to organic compounds. Number 3, bound or precipitated in the inorganic salts. For example, calcium phosphate is the major salt of bones and teeth. I previously mentioned that the calcium atom may lose its outermost 2 electrons, the valency electrons, and become a charged calcium ion. Several proteins, for example, calcium modulated protein or calmodulin, can bind to 4 calcium ions and gets activated. Intermolecular forces such as ionic bonds, hydrogen bonds, and van der Waals forces stabilize a calcium ion within the binding pocket of calmodulin, and in that binding pocket, the calcium ion is surrounded by amino acid molecules. Here in the middle panel diagram is showing the coordination bond distance in angstrom between the oxygen atoms of the amino acid in the metal binding pocket of calmodulin and the calcium ion. How iron exists in the body? At physiological pH, iron exists in the oxidized ferric state. To be absorbed, iron must be in the ferrous state or bound by a protein such as transferrin. As I mentioned previously, metal ion, for example ferric ion, bound to a protein, for example transferrin, is surrounded by molecules or ions of the amino acids present at the binding site of the protein, which forms a coordination complex. So at the center of a coordination complex, there is a metal ion surrounded by atoms, molecules or ions of the amino acids of a protein, which are called ligands. Now ferric ion has a coordination number 6, which means 6 water molecules can surround a ferric ion. Ferric ion couples to transferrin only in the company of an anion, usually a carbonate ion with a coordination number of 2, and this serves as a bridging ligand between metal and protein, and also excludes 2 water molecules from the coordination sites. The remaining 4 coordination sites on ferric ion are provided by transferrin protein residues, a histidine nitrogen, an aspartic acid carboxylate oxygen, and 2 tyrosine phenolate oxygens. Thus, ferric ion forms a coordination complex by atomic interaction with the surrounding amino acids. Now, this is very important because this will explain how a drop in the pH in the surrounding solution can cause conformational change or change in the coordination sphere, which eventually release ferric ion from the transferrin. How iron is transported in the body? Iron is an essential biometal, but free iron can promote free radical formation resulting in oxidative damage. Therefore, iron is transported in a redox-safe state by transferrin and iron transporter serum glycoprotein. Under physiological conditions, almost all iron in the circulation is bound to transferrin. Circulatory transferrin secreted by the liver harvests iron from the intestinal luminal epithelial cells and deliver it to different organs. Iron-saturated transferrin binds to the dedicated transferrin receptor at the luminal surface of the endothelial cells and internalized by endocytosis. Once inside, the endosomal transferrin may release its cargo due to a drop in the endosomal pH to 6 and recycle back to the luminal surface, or it may undergo endosomal transcytosis and release to the basal surface. From there, the iron is transported to different cells in the underlying tissues. At this point, I'd like to discuss about the cholinergic hypothesis of Alzheimer's and the role of calcium in signal transduction. A cholinergic neuron is a nerve cell which mainly uses acetylcholine as neurotransmitter. Acetylcholine was the first discovered neurotransmitter. It acts at various sites within the central and peripheral nervous system, where it functions as a neurotransmitter and as a neuromodulator. The activity of choline-acetyl transferase, an enzyme responsible for the synthesis of acetylcholine, was greatly reduced in the amygdala, hippocampus, and cortex in the Alzheimer brain, along with the reduction in the concentration of acetylcholine. Therefore, it was suggested that hypofunction of cholinergic neurons may be associated with the pathogenesis of Alzheimer. An acetylcholine receptor is a G-protein coupled receptor. Upon binding to acetylcholine, it activates a cascade of downstream signaling pathways which involves recruitment of trimeric G proteins and subsequent release of calcium from the internal storage. This figure is a schematic representation of GPCR signal transduction and GIRK channel activation. GIRK is a G protein coupled inwardly rectifying potassium channel which is present on the membrane. Agonist binding activates GPCR, which then triggers recruitment of trimeric G proteins. As a result, GDP gets hydrolyzed to GDP and causes release of activated G proteins. The activated G beta gamma then binds to the GIRK channel and activates the channel. As a result, potassium enters inside the cell and causes hyperpolarization. The implication of this signal transduction is depicted below. A representative current clamp recording of spontaneous action potentials from an sinoatrial node cell treated with 1 millimolar isoproterenol or acetylcholine is shown here. Isoproterenol is a medication used for the treatment of bradycardia and heart block. It is a non-selective beta adrenoreceptor agonist along with just like epinephrine, and it also acts as a GPCR. As you can see here, that treatment with isoproterenol caused regular interval action potentials. But when acetylcholine was added, the GIRK channels activated, and that caused influx of potassium ion, which hyperpolarized the cell membrane and lengthened the interval between cardiac action potentials, which eventually resulted in slowing down the heart rate. Similar to the GIRK, there are also voltage-gated calcium channels, known as voltage-dependent calcium channels, which also works on the same principle. In the previous slide, I mentioned how GPCR signaling can activate and open membrane-bound ion channels. In this slide, I'll discuss how GPCR signal transduction causes release of calcium ions from the internal storage, mainly endoplasmic reticulum. As you can see here, agonist binding to GPCR, for example, muscarinic receptor, causes conformational change and recruits trimeric G proteins. Activated G-alpha then binds to its effector and activate adenylyl cyclase, which generates cyclic MP. The increased cyclic MP then binds to protein kinase A and activates the kinase, which in turn activates calcium ion channels present on the membrane, and that causes influx of calcium ion inside the cell, increasing the cytosolic calcium ion concentration. In an alternative pathway, the G-alpha protein can activate phospholipase C beta or PLC, which in turn breaks down membrane phospholipids PIP2 or phosphatidyl inositol 4,5 bisphosphate to diacylglycerol or DAG and inositol 1,4,5 triphosphate or IP3. IP3 is a second messenger. The IP3 can bind to its receptor inositol triphosphate receptor or IP3R, which is a membrane glycoprotein complex acting as a calcium channel. Binding of IP3 to IP3 receptor activates the channel and causes influx of calcium ion from endoplasmic reticulum to the cytosol. The increased cytosolic calcium then binds to the calcium binding protein, for example, calmodulin, and causes downstream signaling. In this section, I am showing you a time-lapse live cell imaging video in which the muscarinic acetylcholine receptors in the HEC293 cells were activated by muscarine treatment, and as a result, there was a dynamic release of intracellular calcium ions, which was then characterized. The green color in this video indicates the fluorescence intensity of a free calcium ion indicator, FLUO4, that exhibits an increase in the fluorescence intensity due to calcium ion binding. The cells were live imaged at 2 seconds interval, and the dynamics was plotted in the graph on the right panel, represented by red line. Let me play the video. Now, in my cell communication and signaling paper, I have explained the temporal nature of this calcium release response. You may have noticed that there is an immediate calcium release response which occurred within seconds to minutes, represented by this gray colored rectangle here, and this was followed by a sustained and delayed calcium release response occurred from 2 to 5 minutes, which was represented by the magenta color rectangle here in this graph. Let me play the video one more time. In my paper, I explained that the immediate calcium release response was due to opening of the plasma membrane-bound calcium ion channels, whereas the delayed response was due to endoplasmic reticulum-mediated calcium release response. This conclusion was drawn based on experiments using a G protein deleted cell line and an inhibitor of IP3 receptor. You can see in this graph that the immediate response was abolished in 6 G protein deleted HEC293 cell line, and this is represented by the green line here in this graph. Also, the delayed response was abolished when the cells were treated with 2-APB, which is an IP3 receptor inhibitor, and this is represented by the blue line here in this graph. In this slide, transmission electron microscopic images of a human liver cancer-derived cells known as HEG2 are shown to highlight the intracellular structures of a cell. For example, the nucleus, mitochondria, and endoplasmic reticulum. This is to indicate where calcium is stored inside the cell. The cells were grown on a nitrocellulose membrane for electron microscopic preparation. Inside the cells, calcium is stored in the endoplasmic reticulum, as I mentioned before. The ER structures are shown here as elongated tubular structures. In addition, calcium is also stored in the mitochondria, which are round globular structures. Furthermore, calcium is also stored as bound to cytosolic calcium buffer proteins or calcium binding proteins, for example, calmodulin. Cells keep their internal calcium levels 1,000 to 10,000 times lower than the calcium levels in the blood or interstitial fluid. Thus, when an ion channel opens in the plasma membrane, then calcium ions immediately enter the cell by passive diffusion. The differentially abundant concentration of calcium, magnesium, potassium, zinc in the context of intra or extracellular is represented by bigger fonts. So, sodium and calcium concentration in the extracellular fluid, for example blood, is much higher than inside of the cell. On the other hand, potassium, magnesium, and zinc concentration is much higher inside the cell than it is outside the cell. As I mentioned before, that inside the cell, free calcium is maintained at a low level. Thus, when calcium is released inside the cell, it can interact with calcium sensing proteins in the cytosol and trigger different biological effects. Calmodulin acts as an intermediary protein that senses calcium levels and relays signal to various calcium-sensitive enzymes, ion channels, and other proteins. Calmodulin is a small dumbbell-shaped protein composed of two globular domains connected by a flexible linker. In this video, I have created an animation showing the conformational change of calmodulin following 4-calcium ion binding. This animation was based on the PDB entries which shows crystal structure of apo and holo calmodulin, and the graphics was prepared by UCF Chimera software. Now I reach to a point where I feel comfortable to explain different signaling pathways that activates CAMKK2 and CAMK4 kinases, which are subject of my study. As you can see in this figure, the calcium calmodulin activated protein kinase 2 or CAMKK2, a serine 309 kinase, is a very complex enzyme which is multiple functional domain. N-terminal ATP binding domain, a central protein kinase domain, and a C-terminal autoregulatory or calmodulin binding domain. Multiple protein kinases, for example GSK3, CDK5, and protein kinase A, regulates CAMKK2 function by activating or deactivating phosphorylation at different sites. Again, it is interesting to note that important cell signaling pathways which are associated with different diseases, including neurodegenerative disease, for example, Wnt signaling pathway, insulin signaling pathway, renin signaling pathway, and HOX signaling pathway, controls GSK3 function. Calpain signaling pathway controls CDK5 function, and cholinergic signaling pathway controls protein kinase function. All of them act as an upstream kinase regulating CAMKK2 function. Once activated, CAMKK2 can phosphorylate multiple downstream kinases, for example CAMK1, CAMK4, AMPK alpha, and SORT1. Each of them has their own signaling pathway, which then controls different cellular function. It is important to note that the cholinergic signal transduction pathway can control directly CAMKK2 function. For example, neurotransmitter acetylcholine, when binds to G protein coupled receptors, for example muscarinic receptors, it can cause recruitment of G proteins, which then gets activated. The activated G proteins can then open the voltage dependent calcium ion channels, causing influx of calcium inside the cell. Or it may cause formation of second messenger IP3, which then bind endoplasmic reticulum bound IP3 receptor and open the ion channels, causing release of calcium inside the cytosol. Thus, increased cytosol concentration of calcium then activates calmodulin, which binds to CAMKK2 and activate its serine 309 kinase function. So now we have enough background information to realize the importance of studying CAMKK2 in neurodegenerative diseases. Towards that goal, I decided to identify novel CAMKK2 targets using a proteomics approach, and the findings led to the discovery of CAMKK2 as a regulator of transferrin trafficking and phosphorylation, which in turn identified phosphorylated transferrin as a potential biomarker for Alzheimer's. These findings were reported in my Frontiers paper, and in the next couple of slides, I'll briefly highlight some important data. To identify novel CAMKK2 targets in primary neurons, I used a siRNA-based approach and knocked down CAMKK2 in cultured primary adult rat dorsal root ganglion neurons. The proteins from the control and the CAMKK2 knockdown neurons were then separated by a two-dimensional gel electrophoresis system. In this system, the first dimension separation was based on the protein's net charge, and this process is called isoelectric focusing. The focused proteins were then separated in the second dimension SDS page based on their mass. The two-dimensionally separated proteins appear as distinct spots in the SDS polyacrylamide gel, which are visualized by fluorescent staining. As you can see that a 75 kilodalton protein was found dramatically different in its appearance when compared between the control and the CAMKK2 knockdown neurons. Subsequently, this band was excised, gel excised, and digested by proteolytic digestion, followed by mass spectrometry, which identified this differentially abundant protein as transferrin. Following the identification of transferrin as a differentially abundant protein under CAMKK2 deficient condition, it was necessary to use an antibody-based assay to validate the mass spectrometry-based findings. Towards that goal, the proteins from the control and CAMKK2 knockdown neurons were focused and immunoblotted using anti-CAMKK2 antibody. As anticipated, it was found that the pH-3,4 fraction of transferrin was dramatically reduced in CAMKK2 knockdown neurons compared to the control. Next, a CRISPR-Cas9-based CAMKK2 knockout HepG2 cell line was created to further validate these findings. Isoelectric focusing and immunoblotting of the proteins derived from CAMKK2 knockout HepG2 and control cells revealed a dramatic reduction of pH-3,4 fraction of transferrin under CAMKK2 knockout condition compared to the wild type. Thus, CAMKK2 deficiency significantly reduced a pH-3,4 fraction of transferrin, which was independently validated using rodent neurons as well as human cell lines. Next, it was found that CAMKK deficiency caused abnormal transferrin trafficking. In a live cell imaging-based experiment, fluorescent conjugated transferrin uptake was measured using CAMKK2 knockout HEC293 cells. The HEC293 knockout CAMKK2 cells were generated by CRISPR-Cas9 technology. CAMKK2 knockout and wild-type HEC293 cells were transfected with RFP conjugated transferrin receptor protein and then treated with FITC conjugated transferrin. The cells were live imaged at different time intervals using confocal microscope, and the membrane-bound transferrin was quantified. The relative amount of transferrin associated with the membrane was found significantly high under CAMKK2 knockout condition compared to the wild type. This indicated a trafficking defect under CAMKK2 deficient condition. Following multiple validation of CAMKK2 mediated regulation of transferrin trafficking and phosphorylation, I decided to use a transgenic CAMKK2 knockout mouse model for further validation. Snap-frozen CAMKK2 knockout mouse tissues, including brain and liver, were provided by Dr. Umashankar's laboratory, Indian University, USA. Here in this slide, on the top panel, fluorescent images, CAMKK2 expression in different parts of the mouse brain is shown using CAMKK2 promoter trapped GFP reporter mouse. Based on this data, CAMKK2 and transferrin expression was checked by immunoblotting in different parts of the mouse brain. Here, these immunoblots are showing that CAMKK2 knockout mouse DRG tissues exhibited significantly reduced transferrin. In contrast, transferrin content was dramatically increased in the CAMKK knockout mouse cerebellum tissues and olfactory bulb tissues. Immunoblotting-based quantification revealed that CAMKK2 deficiency significantly reduced transferrin content in the olfactory bulb, cerebellum, cortex, and liver tissues in CAMKK2 knockout mice compared to the wild type. It was also found that in majority of the tissues, the negatively charged pH 3 to 4 fraction of transferrin was dramatically reduced. Based on these findings, it was hypothesized that dysregulation of calcium in Alzheimer's leads to aberrant CAMKK2 signaling, which in turn alters the phosphorylation, that is a pH 3-4 fraction containing phosphorylated residues, and turnover of transferrin during the development and progression of Alzheimer's. It was further hypothesized that the aberrant phosphotransferrin profile that is detected under dysfunctional CAMKK2 condition will be reflected in the serum and cerebrospinal fluid of Alzheimer's patients and therefore can be developed as a potential biomarker for AD. These deductions were further validated using a triple transgenic mouse model of Alzheimer's, commonly known as 3xTG-AD, which was kindly provided by Dr. Ben Albensi's lab. In addition, serum and CSF samples obtained from early and late onset Alzheimer's patients as well as matched unaffected controls were used. These samples were used to further validate the proposed phosphotransferrin-based Alzheimer's biomarker. In this slide, the top left panel is showing diagrammatic representation of the deglycosylation strategy to remove the N or O-linked glycans from transferrin before isoelectric focusing. The deglycosylation of transferrin improved the focusing resolution. The middle panel immunoblots are showing charge and mass separated fractions of transferrin in the CSF and serum samples from a EOAD or early onset Alzheimer patient and the matched unaffected control. The bottom panels are showing relative amount of the pH 3-4 fraction of transferrin in the CSF and serum samples from early and late onset Alzheimer patients. Now I will discuss the findings in my cell communication and signaling paper entitled CAMKK2 CAMK4 signaling regulates transferrin trafficking, turnover, and iron homeostasis. Now first I like to highlight the graphical abstract summarizing the main findings. This study involves the use of a CAMK4 knockout mouse which showed loss of CAMK4 differentially affected transferrin turnover and iron content in a tissue-specific manner. The second part involves use of multiple CRISPR-Cas9 mediated gene-deleted HEC293 cell-based study, which showed that loss of CAMKK2 or CAMK4 or double knockout condition affected uptake of transferrin and endoplasmic reticulum-mediated calcium ion homeostasis, as well as it affected the dynamics of transferrin and transferrin receptor associated protein complex during trafficking. As I mentioned previously, that activated CAMKK2 exhibits kinase activity to three well-characterized downstream effector kinases, CAMK1, CAMK4, and AMPK. So the inevitable question was, is any of the CAMKK2 downstream kinases involved in the regulation of transferrin trafficking and phosphorylation? Now, working with three kinases would be a monumentous task and requires a huge resource which I did not have. So I made a calculative guess based on the gene expression pattern of the effector kinases in the brain. A comparative analysis of the gene expression of CAMKK2 downstream effector kinases revealed that CAMK4 has overlapping expression pattern with transferrin receptor, and CAMKK2 in different parts of the brain. These bottom panel images are showing expression of transferrin receptor, CAMKK2 and CAMK4 in different parts of the mouse brain. Based on these images, it is obvious that all these genes are highly expressed in different parts of the mouse brain, specifically in the cerebellum region. Using CAMK4 knockout mouse, it was found that loss of CAMK4 tissue-specifically altered both transferrin and iron content. The top left immunoblots showing the absence of CAMKK2 or CAMK4 in CAMKK2 or CAMK4 knockout mouse tissues. Immunoblotting-based quantification of transferrin was performed using different mouse tissues, including brain and liver. Immunoblotting revealed that both transferrin and transferrin receptor content was significantly increased in the cerebellum of CAMK4 knockout mouse compared to the wild type. In contrast, both transferrin and transferrin receptor content was significantly decreased in the CAMK4 knockout mouse liver tissue compared to the wild type. Next, the iron content was measured using a snap frozen mouse tissues. It was found that the iron content in the cerebellum and liver of CAMK4 knockout mouse was significantly higher compared to the wild type mouse, whereas the iron content in the cortical tissues exhibited no significant difference. The altered iron content matched to the altered transferrin and transferrin receptor content in the corresponding tissues in CAMK4 knockout mouse. Next, isoelectric focusing was performed to see if post-translational modification of transferrin was affected due to loss of CAMK4, as previously observed under CAMKK2 knockout condition. Isoelectric focusing following SDS page and immunoblotting revealed dramatic difference in transferrin charged fraction under CAMK4 knockout condition in both liver and cerebellum tissues. Relative quantification of the charged fraction revealed significant reduction of negatively charged fraction of transferrin in both cerebellum and liver tissues in CAMK4 knockout mice compared to the wild type. Thus, the observed effect of CAMK4 loss on transferrin phosphorylation and transferrin turnover was similar to that of CAMKK2 loss, but the tissue specificity was slightly different. Next, I have generated multiple CAMKK2, CAMK4, and both CAMKK2 and CAMK4 deleted HEC293 cell lines to study transferrin uptake and to further validate the CAMK4 knockout mouse-based observations. Treatment of wild-type CAMKK2 knockout, CAMK4 knockout, and double knockout HEC293 cells with 25 microgram per ml transferrin exhibited dramatic increase in transferrin uptake under CAMKK2, CAMK4 loss of function condition. These results reinforced the previous findings that CAMKK2 and CAMK4 signaling regulates transferrin uptake. In an alternative approach, the HEC293 cells were transiently transfected with RP-tag transferrin receptor, and then the cells were incubated with fluorescent conjugated transferrin to study the transferrin uptake. The cells were live imaged, and then the membrane-bound or membrane-retained transferrin was quantified. This quantification revealed that significantly increased amount of transferrin was associated with the membranes in CAMK4 or double knockout cells compared to the wild-type cells. These datasets represents another line of evidence that loss of CAMKK2 and CAMK4 signaling affects transferrin trafficking and uptake. The two-dimensional blue native page electrophoresis was adopted to study the protein-protein interactions involving CAMKK2, CAMK4, transferrin, and transferrin receptor during transferrin trafficking. In blue native page, the proteins and multiprotein complexes are separated under native conditions in a gradient first dimension gel. Subsequently, the proteins and multiprotein complexes are denatured and reduced in the first dimension gel strip and subjected to electrophoretic separation in the second dimension SDS page. As a result, the component monomeric proteins will appear on a vertical line in the second dimension corresponding to the multiprotein complexes separated in the first dimension. The right panel immunoblots are showing transferrin receptor and CAMKK2 associated protein complexes under different treatment condition. In the top panel immunoblot, CAMKK2 appeared as a 146 kilodalton protein complex as well as a very high molecular weight protein complex. These distinct protein complexes are absent in CAMKK2 knockout cells as anticipated. Interestingly, transferrin receptor also appeared as multiple protein complexes ranging from 480 kilodalton to a very high molecular weight. The high molecular weight transferrin receptor complex vertically aligned with CAMKK2 complex, indicating that these two proteins may be associated in a single protein complex. Interestingly, when transferrin was added to the media, the transferrin receptor associated protein complexes shifted to a relatively higher molecular weight region. This indicates recruitment of potential interacting proteins. In addition, under CAMKK2 deficient condition, this relative shift of transferrin receptor complexes to high molecular weight region under transferrin treatment was lacking. This indicated that CAMKK2 loss may have affected recruitment of interacting proteins to the transferrin receptor associated complex during the trafficking process. This is a very complex slide, so I'll spend a considerable amount of time explaining the figures. On the left panel, there is a series of immunoblots which represents the relative position of transferrin receptor associated protein complexes during transferrin trafficking. 4 different types of cells are presented here. The top panel is wild type cell, and it is showing transferrin receptor associated protein complexes under no transferrin and transferrin treatment condition. And the double-headed arrows are indicating the relative shift of the protein complexes during trafficking. The CAMKK2 knockout, CAMK4 knockout, and double knockout cells are each surrounded by a colored rectangle, so they can be easily compared. As you can see that under CAMKK2, CAMK4 or double knockout condition, the relative shift of transferrin receptor associated protein complexes under transferrin treatment condition was hampered or abolished. This indicated a defect in the recruitment of interacting proteins during the trafficking process. On the right panel, there are some pseudo-colored immunoblots, and these immunoblots were generated by superimposing transferrin receptor associated complexes and transferrin associated complexes to show relative shift of both proteins during trafficking. As you can see that on the top two panels, both transferrin and transferrin receptor associated protein complexes shifted to higher molecular weight region following transferrin treatment. And as previously showed, the relative shift of both complexes, transferrin and transferrin receptor associated complexes, were hampered under CAMKK2 knockout condition. Similar observations were made under CAMK4 knockout condition and double knockout condition, but one interesting difference is that the transferrin associated complexes moved slightly different from CAMKK2 knockout condition. Thus, the overall data indicates a very complex dynamics of recruitment and de-recruitment of protein complexes under CAMKK2 or CAMK4 or double knockout condition. Now, this is a very complex set of data. Therefore, I'll spend a considerable amount of time explaining the details. Overall, this dataset indicates that CAMKK2, CAMK4 signaling regulates endoplasmic reticulum-mediated calcium ion release response during cholinergic signaling. The top panel figure was presented before, but I'll explain it again for your convenience. The left panel line graphs are indicating intracellular calcium release response during muscarinic activation. Different CRISPR-Cas9 based gene deleted HEC293 cell lines were used, and these are 6G protein deleted HEC293 cell designated here as G60. In addition, CAMKK2, CAMK4 and both CAMKK2 and CAMK4 deleted HEC293 cells were also used. On the top panel line graph, the red line represents intracellular calcium level following muscarine treatment. You can see that immediately after muscarine treatment, there is a sharp increase in calcium ion concentration, which dropped slightly and then maintained at a steady level up to 5 minutes. Now, the use of G60 cell combined with the IP3 receptor inhibitor helped me to dissect this calcium release response into two components. One, immediate response, which was designated as plasma membrane mediated calcium release, and the delayed response, that is from 2 to 5 minutes, was designated as endoplasmic reticulum mediated calcium release. Let me explain the basis of this explanation. As you can see that the muscarine induced calcium release response in G60 cell was presented here as a green line. Under G protein deleted condition, the immediate response was completely abolished, but the delayed response was sustained. Therefore, it was explained that the immediate response was G protein mediated, whereas the delayed response was possibly due to release of calcium from intracellular storage, for example, endoplasmic reticulum. Next, the blue line represents muscarine-induced calcium release response under G protein deleted as well as 2-APB treated condition, and 2-APB is a IP3 receptor inhibitor. Under this combination, the delayed response was again completely abolished. This provided support that the delayed 2 to 5 minute response was due to endoplasmic reticulum mediated calcium ion response, release response. Now, interestingly, the delayed response was found completely abolished under CAMKK2, CAMK4, and both CAMKK2 and CAMK4 knockout condition. And this provided experimental support that CAMKK2-CAMK4 signaling regulates endoplasmic reticulum-mediated calcium release response during muscarinic activation or cholinergic signaling. The right panel images are actually showing the different data points in terms of a scatter plot and the statistical analysis of the data set. Now, in this live cell imaging video, I'm showing you the calcium release response in the wild type and CAMKK2 knockout cells following muscarinic activation. The cells were imaged at 2-second interval, and the green color is indicating the fluorophore intensity change, which corresponds to the free calcium ion concentration. Let me play the video. Now, one more time. At this point, the muscarine was added inside the cell, and you can see that there is immediate calcium release response, which was plasma membrane mediated calcium release. And then this response was sharply declined in the CAMKK2 deficient cells, which indicated that the delayed response was abolished, which was equivalent to endoplasmic reticulum mediated calcium release response. Now, this is another very interesting data set which indicates defective calcium signaling during transferrin trafficking in CAMKK2 or CAMK4 deficient cells. In this set of experiments, HEC293 and CAMKK2 or CAMK4 deficient cells were treated with 25 microgram transferrin per ml, and then the cells were live imaged for fluorophore intensity change. The blue line indicates the intracellular calcium level following transferrin treatment in the wild type cells. The red, green, and magenta colors are representing CAMKK2 or CAMK4 deficient conditions. It is interesting to note that following transferrin treatment, there is a sustained and steady increase of calcium, which was maintained for a prolonged period of time. But under CAMKK2, CAMK4, and double knockout condition, this response was delayed, and the intracellular free calcium level was significantly lower at different time points. Overall, this dataset actually indicates that CAMKK2 or CAMK4 loss cause a defective calcium signaling during transferrin trafficking, which may be associated with the altered transferrin turnover inside the cell. Now, in conclusion, it can be said that this study provided a link between intracellular calcium and iron homeostasis, both regulated by CAMKK2 and CAMK4 signaling, which is vital for cell function and survival. The findings provided here may support an explanation for the behavioral abnormalities observed in both CAMKK2 and CAMK4 deficient mice, and future studies must prioritize CAMKK2, CAMK4 signaling mediated transferrin trafficking in different parts of the brain to understand iron overloading mediated neurodegeneration and cognitive malfunction. Thanks for listening, and if you have any question, then you can email me. My emails are provided at the end of this lecture. Thank you once again.